It is widely believed that Kawasaki disease is of an infectious etiology. After the emergence of MIS-C, the view that Kawasaki disease is also caused by a virus has been gaining momentum. Our group is seeking Kawasaki disease virus. Force of infection, or lambda, is defined as the rate with which an individual is infected with a microbe in a year. Lambda can be estimated from the ages of patients. The geographical distribution of force of infection is identical between Kawasaki disease and pediatric viral infections. In addition, this distribution is similar to that of total fertility rate. These findings indicate that the transmission of Kawasaki disease and viral illnesses are more intense where there are more children in a family. This suggests that the agents for viral infections and Kawasaki disease are transmitted most effectively between siblings. The incidences of Kawasaki disease, chickenpox, parvovirus, and adenovirus have two peaks in Japan, one in June and another in December or January. Hand, foot, mouth disease and herp angina also show bimodal seasonalities in Okinawa. These findings suggest that Kawasaki disease is also a viral illness. These are the annual number of patients of pediatric infections. In most of the illnesses, the numbers fluctuate. Kawasaki disease and exanthema subitum are exceptions. These two shift very smoothly. Mathematically, this smoothness indicates that the causative agents infect the host persistently. Inconsistent, the agent for exanthema subitum infect the host lifelong. We built this differential equation model where V denotes the proportion of individuals who have never been infected with the agent for Kawasaki disease. V is expressed in this differential equation where lambda represents the force of infection. This differential equation can be solved using the data from national census and nationwide Kawasaki disease surveillance. As a result, it is predicted that 88% of children are infected by the agent for Kawasaki disease before their 5th birthday. In addition, only 1% of these infections develop Kawasaki disease. Taken together, the main etiological agent for Kawasaki disease is a virus. Kawasaki disease virus infects the host persistently. Kawasaki disease virus is a common virus. Only 1% of infections with the Kawasaki disease virus develop Kawasaki disease. Common persistent virus in children converge to herpes viridae and anenoviridae, or so-called Teno virus. Teno virus was discovered by Nishizawa et al. In 1997. This group of virus was later given genus name of anenoviridae. This virus infects all the vertebrates. 30 species of Teno virus infect humans. This virus is present in almost all bodily fluid. Almost all human beings are infected with the Teno virus in childhood. Infections with the Teno virus persist. We conducted a prospective metagenomic study to explore Kawasaki disease virus in Osaka Kosei Nenkin Hospital between 2014 and 2015. 11 patients with Kawasaki disease, 11 control subjects with diarrhea, and 11 controls with respiratory infections were enrolled. Each control individual was matched to a patient with Kawasaki disease in terms of age, sex, and time point of sample collection. Serum, whole blood, nasopharyngeal aspirates, and feces were collected from participants. DNA and RNA were extracted and sequenced in Lawrence Livermore National Laboratory. Other microbes, including virus, bacteria, fungus, and archaea, were identified by bioinformatics. Viral DNA and RNA are only a very small portion of the entire nucleotides in biological samples. We extracted viral DNA and RNA carefully, maximally eliminating host and bacterial DNAs. Metagenomic sequencing showed that most of the DNAs in the serum and whole blood collected and pulled from Kawasaki disease were viral. In contrast, most of the DNAs in control subjects were bacterial. Over 95% of viral DNAs in the samples from Kawasaki disease were of Teno Virus 7. 2 of 11 patients with the Kawasaki disease were positive with Teno Virus 7, and their viral loads were very high. In contrast, all of 22 control subjects were negative with Teno Virus 7. Teno Virus 7 was discovered in 2000 and is very rare. To explore the role of this virus in Kawasaki disease, I visited Fabrizio Maggi in Italy, an authority on Teno Virus. We quantified Teno Virus in 57 serum samples collected in multiple hospitals in Osaka between 2002 and 2005. There was a strong correlation between Teno viral load and total bilirubin. That is the most efficient predictor for IVIG resistance. 3 patients were positive with Teno Virus 7, and all of them showed a high viral load. Of note, the patient with the highest Teno viral load and the highest total bilirubin was positive with Teno Virus 7. There was a significant correlation between Sano's score and the Teno viral load. The correlation between IVIG resistance and the Teno viral load was marginally significant, with a p-value of 0.16. The sample size necessary to show a significant correlation was estimated to be 150. A future study should elucidate whether high viral load of Teno virus is the cause or result of Kawasaki disease. Some species of Teno virus may be prone to develop Kawasaki disease, to resist to IVIG, or to form aneurysms. Interaction between Teno virus species and host genotype is an interesting topic for further investigation.